begin.
This is a modeling for process meiosis, and here I have 4 snake-like chromosomes. I have 4 different colors showing us that they have 4 different traits in them. Okay, now I have 2 different lengths here to show the homologous chromosomes. The shorter ones, they are one homologous pair. The longer ones are another homologous pair. In these homologous chromosomes, they have the same traits. For example, if it's a trait for hair, they have the hair trait on the same location responding to each other. That's why they're called homologous pair. Now we start with meiosis process with interphase. In interphase, just like mitosis, each chromosome, so I have 4 chromosomes here, each chromosomes duplicate themselves. So that's what is happening, duplication of chromosomes. So I have chromosome here which has been duplicated. So I have one. This is being made long by my son, so I'm trying to make it shorter now. Then I have my green duplicating, then I have my yellow duplicating, then I have my pink duplicating. So in my interphase, all my chromosomes have duplicated. Okay, so that's all I'm doing in my interphase. In my prophase 1, again like mitosis, most things happen in prophase. In meiosis, we have 2 prophases. That's why I'm going to call it prophase 1 and prophase 2. In my prophase 1, what is happening is just like my prophase in mitosis, the 2 chromosomes are going to attach at the center through centromeres and form sister chromatids. So now I have sister chromatids, 4 chromosomes doubled up, so I have 8 chromatids and they are sister chromatids, just like my prophase in mitosis. Now next step is what is different in meiosis. In meiosis, after they have become chromatids, after they are connected at the center through centromere, there's something happening called crossing over. By crossing over, what I mean is the 2 homologous pairs, so the purple and the blue are homologous pairs, the pink and the green are homologous pairs, they come close to each other. And then they exchange some genetic information. By exchanging, they cross over each other. Okay? Yep, this is how the crossing over takes place. So when they cross over, they cross information, and I'm going to show that by exchanging some of the color from each of the chromatid and putting it on the other. So my daughter Navya is helping me do on the other side. Okay. So now I have the 2 homologous pair, but some amount of information has been exchanged. The purple has some blue in it, the blue has some purple in it, the pink has some yellow in it, and the yellow has some pink in it. And this is known as crossing over, and this is specific to meiosis. It happens in prophase 1. Once they have crossed over, they have exchanged their information. Now we go on to the next process, which is metaphase, just like mitosis. But because I have 2 metaphases, I'm going to call this metaphase 1. And just like my metaphase in mitosis, all that they do is line up. But in this case, the homologous pairs line up adjacent to each other. So this is how it's going to look when they line up. Okay, so they're all lined up in the center of the table. Yep. Once they are lined up, now they are lined up, so metaphase 1 is done. In my anaphase, those spindle fibers come up. So I don't have those up here, but I will have twine for you to show to the other students. Those spindle fibers come up here and pull on the chromatids. Now instead of one chromatid going away, like in mitosis, here the homologous pairs go away from each other. That is the difference. In anaphase and meiosis, the homologous pairs are pulled away by the spindle fibers. So now you see I have two sets here. So basically these are two cells formed. Okay, I have one cell on this side and I have another cell on this side. Now this is my end of anaphase 1. Okay, and in my telophase, these two cells, they form their own nuclear membranes and they share the cytoplasm just like my telophase in mitosis. Now here is the difference. Once this is done, it doesn't stop here. This is just an intermediate situation. In meiosis, it continues. Now what happens in the next is a mixture of prophase and metaphase. So we call it prophase 2 and metaphase 2 together. So prophase 1 or prophase 2, in prophase 2, they are coming together. Each cell is coming in line, so they align themselves. Okay, they were already aligned, but I'm kind of showing they readjust themselves, make sure they are aligned in the middle, and you have those spindle fibers popping up again in each cell, and each of these chromatids are pulled apart in anaphase 2. So basically now each of these chromatids are pulled apart. Navya is helping me again, pulling apart the chromatids. So we are the spindle fibers here. Navya and me are the spindle fibers here. We are pulling it apart. So we have pulled apart the chromatids. So now we have 1, 2, 3, and 4 cells. Okay, so we have, I'm looking for a sketch pen here. There was a sketch pen here someplace. No, I had sketch pen up here. It's missing now. Probably Arjun took it. But the sketch, this is divided. Each one of these is divided into 4 cells. So I have 1 cell, 2 cell, 3 cell, and 4 cell. We started off with 4 chromosomes, doubling up, that is 8 chromosomes, and now I have all the 8 chromosomes up here, but they are in 4 different cells. Okay, so basically my diploid cell has become 4 haploid cells, which means it has half the number of chromosomes that the original cell had. Okay, so I have 4 haploid cells. This is what happens to all sex cells. So all the sex cells in male and female reproductive system are like this. They are only half of what the original number was, and in the process some information has been exchanged. So not each cell or not any one of these cells is similar. Each one of them have a different combination of colors, and that's why we have a couple can make babies who are of different color, different size. They don't look identical. They're non-identical kids because each of the genetic material combination is different. Okay, hope this helps you, and this I'll help you in class 2 to do the same thing with the rest of the students. Okay, thank you, and thank you Navya, and thank you Arjun for helping me out do this demo. Thanks.